it's a real pleasure to have been invited to talk to you. I've been at the Royal Botanic Garden Edinburgh for coming up for 3 years, and it's, as I say, it's a real privilege to be meeting with this particular community of people, some of whom I know will know us well, and others perhaps less so. But I thought I would take the opportunity this evening to share with you a little bit more about our approach to our collections and the role of our collections, focusing on preserved collections. So, as Rich said, I'm responsible not only for the herbarium, but also the library and the archive collections. And we have an amazing team of collections, a collections-based team here looking after those collections. So curating and managing these collections in perpetuity as part of Scotland's national botanical collections. So, as I say, I thought I would give an introduction to the herbarium and the role of herbaria in biodiversity research, including our own here at the Royal Botanic Garden Edinburgh, and then finally end off with some ways in which plant recorders here in Scotland in particular, but in the UK in general, are really helping us to achieve more with what we do in this time of biodiversity crisis. So a herbarium is essentially a museum of dried or dead plant specimens, and traditionally also includes fungi, because when herbaria were established in the 16th and 17th centuries, fungi were regarded as plants or honorary plants. And as per tradition, we continue to curate collections of fungi as well. But I do stress these are dead collections rather than living collections of cultures and things. Here, this is a photograph from inside the herbarium here. Can I see a show of hands just in the audience who has visited us in the herbarium? Oh, yay. Great, so you know us and you probably recognize the scene. The vast majority of our specimens in the herbarium and in any herbarium are flattened vascular plants. There are always some non-vascular plants as well, but the vast majority are flattened, which makes our life relatively easy compared to the curators of other collections, which involve lots of bulky 3D objects. However, we do have our own 3D objects as well. So in this slide, you can see a selection of our other collections, which include a carpological collection of seeds and fruits. It may include parts of the pressed plant material that are impossible to press completely flat. And in that case, they need specialist mounting techniques to ensure that the integrity of the pressed specimen is maintained. We have a wet tissue collection. So this is plant material preserved in preserving fluid. It's a mixture of alcohol and a few other ingredients. And we also have a tissue collection preserved in silica gel. So all of these supplement that vast collection of primarily flattened plants material and fungi. And the objective here is that if any diagnostic characters are likely to be lost in the pressing process, which would make identification very difficult, then the idea here is to maintain that three-dimensional structure so that the important diagnostic characters or any features that a researcher might be interested in are preserved carefully, as I say, either by drying directly or by being preserved in fluid. So the herbarium is actually a lot more than just cupboards full of flattened plant material. Here in Edinburgh, we also have macroalgae, of course, the coastline of Scotland being particularly interesting for these collections. So we think we have about 3 million specimens. We won't know until we finish digitizing them, and I'll get onto that in a moment. Our collections represent several centuries of collecting efforts. Our earliest specimen is from the 17th century, and we accession up to about 30,000 new specimens every year. So Edinburgh is probably among the most active herbaria in the world, in that we're very vibrant and we continue to accession new material all the time. We are the National Herbarium of Scotland, and so not surprisingly, our collection of material from the British Isles is particularly strong, and of course, there's an emphasis on Scotland. So any collecting that happens in Scotland by default will come to this National Herbarium before going on potentially to be shared as duplicates to one of the other main herbaria, particularly BN at the Natural History Museum. So we have over 278,000 specimens for the flora of Britain and Ireland, which includes a great many type specimens as well. And obviously, not surprisingly, the vast majority of flowering plants and specimens from Scotland. That said, though, we are actively developing the non-flowering plant collections, and the curator of the cryptogamic collection is actually here today, my colleague Helene Plaisier. Helene's in the back there. All of these collections from Scotland and Britain and Ireland can be found online because they've all been databased, and the vast majority of them have been imaged as well. So they're easy to find on our online catalogue. I mentioned type specimens, and I know this audience will appreciate the significance of this. All of our type specimens are digitized. So a type, there are various categories of type specimens, but of course, the type specimen becomes the biological entity against which a name is pinned. When a name is published for the first time, it must have a voucher specimen that is regarded as a type, at least one, if not many. And due to the significance of type specimens in nomenclature and taxonomy and their value to the whole classification process, we curate them slightly differently in that we put them in these elegant red folders. So opening a herbarium cabinet, it's really easy to spot those significant type specimens. This makes life much easier for researchers who are busy working with the collections and will ordinarily start with the type specimen before looking at others. But it also, in a worst case scenario, if we really had to prioritize, we would always aim to preserve or save the red type folders, unfortunately, before anything else in the collections. Here in Edinburgh, we've also innovated recently by introducing some lovely purple folders, and these are what we refer to as genome types. I'm not aware of other herbaria doing this, but we think that the voucher specimens that have donated material or come from the same plant or indeed other organism that has had its whole genome sequenced and become a reference genome. We think those specimens are very important as well. And so we have started preserving or curating those specimens in these purple folders. Again, for research purposes, it can really speed up the research process if you really need to look at the material that lent its genome to be sequenced. So here's an interesting comparison between our earliest specimen, and which was collected in 1697 in the Cape of South Africa, versus the millionth specimen that we digitized, which happened to be a lichen, collected by my colleague Becky Yar, who is one of our resident lichenologists at the Royal Botanic Gardens at Edinburgh. Now, if you look at these two specimens, you might not guess that there's several hundred years between their collecting dates, because fundamentally the practice of preserving material in a herbarium has not radically changed over the centuries. Indeed, the collecting process also hasn't really changed significantly. We still suffer from an image crisis of sometimes being regarded as a kind of Victorian hobby, collecting plant material or fungi. And of course, again, those of you in the room will know that it couldn't be further from the truth. Plant collecting to preserve voucher specimens in a herbarium continues to be a very intrepid pursuit and sometimes potentially life-threatening. So it's quite the opposite of being a hobby. But the practice of drying and preserving these specimens in perpetuity hasn't adjusted much. When I'm giving tours of the herbarium, I like to point out two things though. One of which is the barcode, which has revolutionized the way that we can curate and manage collections like this, because we're able to track a specimen in the building. As long as it's got a barcode, we know exactly where it is, or at least where it should be. This makes life a lot easier for us in terms of collections management and curation. But the other massive innovation in the way that we approach collecting, and indeed the way we approach data, are the fact that we now have georeferences on our specimens, and the accuracy with which we can pinpoint exactly where a specimen was collected has massively changed with the availability of satellite data. And so that would be, I would say, easily the most significant innovation that's happened in the centuries between these two specimens, because now we wouldn't even leave the Institute without a Google map with localities that we intend to visit, all the satellite data that goes into the planning process, and then of course results in this very, very accurate georeferenced location for each specimen. And that's one of the reasons why herbaria are now proving to be extremely important in understanding patterns in biodiversity. And I'm going to get onto that in a moment. Let me just check my time. So not that much difference in collectors going out looking for particular plants or fungi, seeking a particular population, looking to gather evidence of biodiversity in the wild, and then preserving these specimens in perpetuity as a record of that biodiversity, both in space and in time. But as I say, some important innovations that actually do separate these centuries of endeavor. And one of those additional innovations that has happened in the last few decades rather than centuries is that we now have an extended specimen concept that includes a digital specimen. So rather than simply having that physical specimen, which might have an additional seed specimen or may have additional material that we curate slightly differently, perhaps spirit collection, we now tend to have a digital specimen. Indeed, we are more than halfway through creating these digital specimens for the Herbarium in Edinburgh. It's powerful because we image the specimens at very high relatively high resolution. It gives us the opportunity to look at specimens in a great deal of detail on a screen, while perhaps also looking at the physical specimen. And every specimen will allow you to zoom into the equivalent of looking at it through the hand lens, so 10 times hand lens. And you can do this from your desktop for free as long as your internet connection is fast enough. And this is important because it has democratized access to herbaria. We're not the only ones digitizing, and really changed the way we can prepare for collecting and enhance our research as well. So as I say, all of these specimens are being made available for free online to anyone with an internet connection. The digital specimen, of course, requires a team of expert digitizers, and this is a snapshot of our digitizing team in action here in the top floor of the herbarium, where we have a very well-oiled machine in terms of the workflow that supports digitization. So it's summarily, it's a two-step process that involves humans capturing some data that is attached to that barcode, and then humans capturing this high-resolution image with a customized camera setup, which you can see in action here. And so our digitizing team are systematically going through the herbarium, creating a digital specimen that's a mirror of every one of the physical specimens in our collection. We're making those digital specimens available, as I say, for free online. And this, I really like to observe our progress in real time, because we're uploading these specimens on a very regular basis. And this particular map will show you the distribution of our specimens that have been digitized. So these are the ones that have their own digital surrogates already, as represented on GBIF. So you can see that we've imaged, or we've shared to GBIF over 1.5 million digital specimens. And this is the distribution of those. So this is not yet our fully digitized herbarium, and it will be interesting to see how that map changes as we continue to digitize. But it gives you an interesting snapshot. And again, if you know our collections, you'll know that we have particular strengths in the parts of the world that are represented here by the darkest orange colors. And there are multiple ways of finding our specimens online. You can go to our website and search directly into our catalogue. Just the Herbarium Edinburgh catalogue. We aggregate to a number of different places, though, so it's also possible to find our specimens amongst others via some UK dedicated resources, including the Disco UK platform and the MBN Atlas as well. And then finally, globally aggregated along with Herbaria around the world that are also sharing their digital specimens to GBIF, which is probably one of the easiest to search. So we are increasing the ways that we share these digital specimens all the time, and making sure that they're as accessible and easily accessed as possible. So it gets our specimens and their data way beyond the walls of the herbarium in which they're kept. And I should just say that we also, as well as having this crack team of digitizers here in the herbarium, we also have an online volunteer community who are helping us to transcribe some of the data from the specimen labels, or to check that automatic transcriptions have been accurately recorded. And so we tend to have a variety of citizen science projects on the go at any one time through this amazing online community who are distributed around the world. Also, we're sharing regular updates on our progress to create this digital herbarium. So I really recommend taking a look at our Botanic Stories website, where you'll see regular updates from the digitizing team, but you'll also find lots of other fantastic stories about what's going on behind the scenes, often here at the Botanics. I want to pause then, having considered our wonderful herbarium here at the Botanics, established in the mid-1800s, like many herbaria were, built up over the years to reflect the collecting activities of our staff and associates, and indeed the community in Scotland as well, because we don't just take specimens that have been collected by RBGE associates. We will also take specimens that are collected by non-associates. And we also receive duplicates in exchange from other herbaria. So there's this very robust network of herbaria around the world. And so I'm going to shift focus now from Edinburgh to a broader kind of global view of herbaria and their role in biodiversity research. So we're fortunate in the community of Herbaria natural science collections to have an index of the world's herbaria, which currently comprises over 4,000 across the globe. That's not necessarily exhaustive. Index Herbariorum gets new herbaria being added every year. But it's, I think, sometimes pretty impressive to look at this map and look at that distribution of these preserved plant collections that have been serving communities of specialists for centuries in some cases. And currently, as I say, more than 4,000 herbaria across the globe, an estimated more than 406 million specimens between them, and over 13,000 staff and associates that we know of currently. So information that's painstakingly curated to reflect this endeavor of collecting plant and fungal specimens as a record of biodiversity. Now, you may have seen in the blurb that was circulated for this evening's talk that I consider this tremendous effort, which collectively has provided this incredible reference, but the reasons under which, or the kind of scenarios under which herbaria are established can differ quite markedly. So each herbarium has its own unique origin story of why it was set up and whom who the collectors are who've contributed to it. Generally, herbaria are established to provide a local 3D reference of plants and fungi growing in a particular place, and that will then that purpose for the collection will then serve it. And indeed, for many years, when the discipline of botany was being established, these private herbarium collections also were very, very important tools for plant identification. You can imagine those centuries of collecting efforts resulting in these millions and millions and millions of specimens over 4 centuries. And then you also imagine the changing planet over those 4 centuries. It becomes very obvious that these near continuous records of plant and fungal diversity that have been collected and accurately named by specialists for so long now provide a window not only into contemporary plant and fungal diversity, because we continue to add to these collections, but they actually also provide a near continuous record of biodiversity in the past. And one of the principal purposes that we are now able to use these data for is to understand how plants and fungi have changed over time, both in terms of their distribution, but also extinction and range expansion as well. So those data that we gather on a specimen label, which may seem remarkably similar to those specimens from 300 years ago, continue to be just as relevant now that they were in the past, but also will be in the future for understanding biodiversity change. One of the most important purposes, though, for herbaria is in supporting our description of biodiversity. I like this graphic from a paper by a former colleague, Tuula Niskanen and colleagues, which describes our progress in describing the world's biota. So plants and fungi, plants doing relatively well in terms of, we think we've named about, or nearly 90% of plants that do exist. So we have a kind of Rumsfeldian known unknowns. And we know what the challenge is that remains to describe that remaining biodiversity. But unfortunately, we also know that a great deal of plants are threatened with extinction. And if you look at the progress, though, compared to, say, vertebrates, where we think we've named about 92% of vertebrates, and also a great deal of them are threatened with extinction, you can see the scale of the challenge that that faces mycologists describing fungal diversity and indeed entomologists describing insect diversity. So we know that we're doing relatively well with plants, but that's not the case for fungi. And so there's this whole other frontier of biodiversity that needs describing, and these collections help with that process. And for plants, we know that about half the plant species that are described every year have actually already been collected and preserved, and they're in herbarium collections. And this gives us a great deal of confidence in terms of, if we make these herbarium collections accessible to the experts who describe this diversity, the rate at which these remaining sort of 10–12% of plant species to be discovered or to be scientifically named, about half of them are likely to have already been collected. But of course, that's not the case with other kingdoms. We also know that making these herbarium collections accessible is really important to support other areas of taxonomy and classification. Indeed, we also know that there's a direct correlation between those specimens being included in a monographic study. So this is a monograph being a very targeted and deep dive into the relationships between species in a group of organisms, be it a genus or family level. And we know that if that hasn't happened, if an expert hasn't written a monograph or provided that level of very detailed study for a group, it's much more likely to become extinct before it gets a name. So again, in that 10 or 12% of species that remain to be named scientifically, the risk is much higher if those specimens of a particular taxon haven't been included in a really detailed study. And so this is also the reason why we need the herbarium specimens that can be included in such detailed study, ideally online, so that there's no physical barrier to access. But we also know that we need the experts. We need taxonomists and systematists who are able to do this very detailed work. And we also know, of course, that, stating the obvious, but without a name, it's impossible to communicate anything about a taxonomic entity, and it's particularly impossible to conserve it. So we really take seriously our role in curating these collections of specimens that can then inform the taxonomic process. Another purpose for which herbaria are now very much celebrated, and indeed museum collections generally, is a new field that's been sort of christened museomics, or indeed herbariomics, which has been made possible by innovations in DNA extraction and sequencing technologies over the last sort of 10–20 years. So it's now possible to get very degraded, but to get DNA from specimens that may be hundreds of years old. And what's quite remarkable about this is that our approach to preserving herbarium specimens, indeed any museum specimens, is pretty much the opposite of what we would do if we were aiming to conserve their DNA. So we collect them and dry them quite slowly, subjecting the DNA to probably a lot of enzyme damage during that process. Historically, the specimens might have been treated with chemicals to encourage preservation, which would also almost certainly have destroyed DNA. And we also freeze the specimens as part of our integrated pest management approaches. So all of these steps in the process of making and preserving a herbarium specimen are actually the antithesis of what we would do to conserve the DNA inside them. And yet, the technologies have become increasingly sensitive, and our chemical means of extracting DNA from these tissues have improved to such an extent that we can now sequence DNA from very old specimens as well as very young specimens. And this enables us to massively increase the number of samples that we can include in tree of life studies. And of course, it's the tree of life that then informs that taxonomy and systematics process that I've mentioned previously. So at Edinburgh and Herbaria around the world, we're hosting very expansive projects and programs of work to sample small amounts of tissue from herbarium specimens without damaging or reducing their integrity and their usefulness as a record of biodiversity, and then using those samples as sources of DNA that can be used in very large tree of life projects, one of which being the Darwin Tree of Life project, which is aiming to sequence whole genomes as well. And you can spot the destructive sampling here, for instance, where a small fragment of plant material has been removed from the specimen. It has damaged the specimen, but it hasn't decreased its integrity or usefulness for science. And this is just one example of many, many studies that are taking this museomics approach these days. I know that, again, in this audience, there's no need for me to convince you that conserving biodiversity and understanding patterns in biodiversity change is a worthwhile endeavor. But there's an increasing amount of evidence that speaks very clearly to the actual monetary value of biodiversity and the risks that we face if we don't conserve it and don't seek to understand changes to biodiversity. So obviously the Dasgupta review some years ago now really illuminated that and put it in fairly crisp terms that were could be understood, as I say, from a financial perspective. That I like to highlight also a report that was done by Deloitte on Australia's biodiversity, where they posed the question, what would it cost if we sought to describe the entire biota of Australia in a single generation of systematists? What would that cost? And indeed, what would it cost if we don't? And so for any of you who are interested in that value of biodiversity and how we communicate it, I strongly recommend this report for some really interesting reading, where it's really spelled out in those kind of black and white monetary terms. And so, as I say, digital herbaria and natural science collections, the physical things, are vital to understanding biodiversity change. They present us with a near complete record of several centuries of collecting pretty much globally, because natural historians and collectors have been intrepid in their collecting efforts to understand Earth's biota. And these specimens are now beautifully preserved in collections around the world. But I like this image because it gives a snapshot of the complexity of, okay, well, we can just digitize them, everything's going to be all right. It's true, digitizing is our absolute baseline, the simplest thing we can do to enhance access and to democratize access to these specimens and the data they contain. But it's a non-trivial task. And so in the final few minutes of my talk this evening, I'm going to just turn my attention to a couple of steps that we've taken very locally here in Edinburgh, in the Herbarium, that are shifting the way we approach day-to-day collections management and curation in response to this biodiversity crisis, because it's absolutely urgent that we understand biodiversity change and continue to describe the Earth's biota in this face of extinction risk and the possibility that it may already be too late to identify and describe some of this diversity. So in 2024, shortly after I joined, we ran a prioritization exercise in our what we call the backlog. So we have a herbarium of about 3 million specimens of mounted, prepared specimens that are accessioned to the herbarium, but we also have thousands of specimens that we've not yet got round to, because the scale of the challenge and the resource available, unfortunately, don't quite line up. This is not unique to Edinburgh. Every collection has a backlog, so it's totally normal. And we've kind of learned to tolerate these backlogs and learn to live with them until the biodiversity crisis, which has reminded us that every one of those records, every one of those specimens might potentially be the missing link in some puzzle that we are aiming to solve. So in 2024, with some of my staff in the Herbarium team, we ran an exercise to pull out those unmounted, unaccessioned specimens from our backlog from biodiversity dark spot regions. Now, biodiversity dark spots are places on the planet that we know have been inadequately described and that are also at the greatest risk of disappearing before we can describe them. So in other words, if there was a global prioritization exercise for natural historians and biodiversity scientists to focus our attention, this is arguably where our attention should be focused. And in just a single exercise, very local, a few members of my team, we were able to identify more than 3,500 specimens from those dark spot countries that arguably should have been prioritized and should already be digitized and available to support biodiversity science across the globe. So that's just one herbarium. And that wasn't even, as I say, it was a relatively straightforward exercise because we know enough about our backlog to be able to find them. But if you imagine the similar process happening in 4,000 herbaria across the globe, it's very likely that the shape and extent and the coloring on this dark spots map might shift simply by looking for specimens that have already been collected, a little bit like looking for those species that have yet to be scientifically described. They've already been collected. And then another thing that we did last year, which I hope some of you joined us for, was after the Scottish Botanists Conference, we held a herbarium open house where delegates who had attended the conference had been in touch with us to say, we would like to come in and look at those unmounted, unaccessioned specimens. And our intention with that was to get the people who know this diversity best to look at some of our British and Irish material to help us identify specimens that might be particularly valuable as new records, new county records. We didn't expect to find new taxa, but we expected to find specimens that have been overlooked and actually turn out to be part of these missing links that can really enhance research. And so we hosted 20 plant recorders from the the BSBI network. And we found 30 new county records in one box of mixed specimens, which was, something that we hadn't expected that level of success. We found specimens dating back to 1800. Again, we didn't know that we had unaccessioned specimens so old. And in total, we were able to fast track 645 specimens to be mounted, barcoded, digitized, accession to the collections, and then therefore available to the people who are really studying and recording biodiversity. And then finally, we're doing a more strategic and scientifically approached gap analysis of the collection. My colleague Marie Briggs is working on this as part of her PhD, based with us in the Herbarium, where we are taking our collections for Britain and Ireland, and we're comparing them to the records that BSBI have gathered as part of their obviously decades of endeavour as well. And we're looking mutually across the two data sets for gaps, because we've already got pilot evidence that suggests that we have specimen data that are not yet in the BSBI database, and vice versa. So we have specimens that may have been collected by BSBI recorders, which can be enhanced by the data in the BSBI database. And so there's this wonderful collaboration between these two long-standing and long-running endeavours to describe the biodiversity of Scotland and the British Isles. And there's opportunities for us to fast-track the way in which we can enhance our specimen data. And so, as I say, Marie Briggs is leading on that. And that's it. So we are responding to the biodiversity crisis by doing more of the same, but also doing some of our things differently, and thinking about our role in this global ecosystem of herbaria somewhat differently. So thank you.